So here's a mic. Okay, I just do it too. Yeah.
Do I start now or 5 minutes? I think.
Yeah, let me check.
Based on the agenda, in 5 minutes.
Okay.
Okay, then let's just ask them on Slack if they are ready.
Yeah.
Okay, Alan, I think they can start.
Okay.
So good evening, everybody. I'm very happy to be here, and I'm talking about exploiting data in medicine. So this is work that I've done at various places that I work at. So first I'm going to tell you about how knowledge is gained in medicine today. So medicine, like many other scientists, people do experiments and they publish papers describing the experimental results. And lots of these papers are published. So I have a graph over there showing the number of papers that are indexed in PubMed starting in 1950 and going up to 2018. So in 1950, there were about 100,000 papers published in the medical area. Last year, almost, well, over 1.3 million. Now, many of these papers don't have anything to do with human medicine. So there are a lot in there, for example, about mice. So sometimes this gets confusing, especially for journalists who are writing for magazines and newspapers. So you might find an article like that that says, you know, this diet is not effective, it causes blood sugar problems in women. What they don't point out there is the experiments were done on mice. So actually that should be causes blood sugar problems in female mice. This doesn't make the same impact. But anyway, there's a Twitter account that always tweets for articles like that in mice. Okay, so once you've taken out the papers that are about mice, about zebrafish, about fruit flies, about all the others, you're left with what actually applies directly to medicine in humans. And those generally happen to be in the form of clinical trials. So that's a sort of typical clinical trial publication where they've taken people who have hepatitis B, they tried 2 treatments on them. One of them has to do with a standard hepatitis B treatment, and for the other one, they give them additional injections of some Chinese medicine herb. And they did that on 120 people, 60 with the old method, 60 with the new method. So the problem with that, of course, is 120 people are not very many. So what also happens is, yeah, generally there are lots of these clinical trials done around, and then they want to know, okay, so how can we get more statistically significant results? And for that, there is a process called systematic reviewing. So that's what a systematic review looks like. And basically, what the authors of this do is they find all the papers that have to do with this particular Chinese herb and hepatitis B. So they found papers describing 35 clinical trials with 3,500 participants. So you see sort of 100 participants per clinical trial, so not very many. But then if you put them all together and reanalyze the data, so do some statistical analysis, you sort of get results on 3,500 participants, which are statistically more significant. Now, a question about this is, how long do you think it takes to write such a systematic review? So from the time when the review is registered, so people decide to do it, until the paper is published. So shout out some numbers in weeks, on average.
2, 12, 50,
20.
67.3. So that was another study done on 192 systematic reviews. So it means it takes about 1.3 years to actually get the information out with more statistically significant information on which treatments are good for specific diseases. So something that we did is we asked ourselves the question, so what if instead of 67.3 weeks, you could do this in 1 second? And we tried. So what we did was to try and extract the key information out of these papers which are describing the clinical trials. And what people working in medicine have worked with for a long time are called the PICO elements, so population, intervention, comparison, and outcome. What does that mean? So here I have some title that I invented for a potential paper. So bioequivalence of aspirin versus vitamin C in children with a headache. So if you want to extract the population, that's basically here the group of people that the experiment was done on. So here children with headache. Intervention is what treatment was tried. The comparison is what treatment was compared with. And the outcome is what was found out. So in this case, that aspirin and vitamin C are the same, but as I say, I just made this title up. So as an example of what we would like titles to look like to make our work easier. So we worked on automating this. Now, of course, the first thing we needed was to know exactly where are the PICO elements in the titles of these articles. So we thought, well, we'll just ask the experts. So we set up some annotation. We had a bit of funding to do this, so we hired 4 medical experts and 2 linguists to annotate the PICO elements in the titles of the papers. And we set up an interface like that, and we set them going. The problem with this was, the first time around, the inter-annotator agreement was so horrible that we couldn't use the results. So it took us 3 iterations of redesigning this interface, always adding additional constraints to try and avoid the annotators doing strange things, so we could get the inter-annotator agreement up to the level that we could actually use those annotations to learn the PICO elements. So in the end, after all of this, we had 1,750 titles annotated with the PIC elements. Now again, 1,750 is not a lot, so we couldn't really do much fancy machine learning, so we decided to take a rule-based approach, so classic natural language processing with some regular expressions. So we first tokenized the titles, then we did a part of speech analyzer, so it found the nouns and prepositions. Then we also had a natural language processing pipeline that was medicine specific, so it found the drugs, the people, and the diseases. And with that, we could then set up some rules, like that one, so for population, where if you have a person and then the word with and disease, then that's a population. So here, children with headache, for example. Of course, our actual rules were more complicated than that, but we got by with about 7 rules for population and 14 rules for intervention and comparison. And with that, we had a decent performance. So what did we do with this information? Well, we could then run it on a few 100,000 clinical trials, publications. So that's an example of one of them. So here you can see the population is in blue, so we have subjects with risk factors for type 2 diabetes. The intervention was metformin, and the comparison was a placebo. What I haven't mentioned yet is the outcome. So it turns out that the people who write the papers tend to not put the outcome in the titles. So actually where we found the outcome was in the conclusion of the abstract. So we basically did some specially trained sentiment analysis on the conclusion to find out if what the paper says in the conclusion is positive or negative. So does the intervention work or not? Of course, we found many more papers with positive sentiment, but that's how publishing works. And then we also extracted automatically the sample size, so the number of people on which the clinical trial was done, because that tells you the power of the result. And then we had a partner, so the TRIP database in the UK. So they took all of the data that we'd extracted from these clinical trials. So what the TRIP database is basically a search engine for medical researchers. So they already index all of the clinical trials. And what they wanted to do was find a better way of displaying the results of the clinical trials. So this is what they came up with based on the information that we extracted. So what you see here is, so basically you type in a query for disease, asthma. It then looks through and sees which interventions have been tested, and it shows the interventions on a plot like this. So here, towards the top, are the interventions that are more likely to be effective. Towards the bottom are the ones that are less likely to be effective. And then the size of the samples grows from left to right. So just by looking at this, you can get an idea within 1 second of which interventions work best for your disease. And you can also drill down. So if you click on one of these bubbles, it shows you the information that it's based on. So that you can click on these individual points and go to the papers that this is based on. So of course we don't say this is as good as something that takes 1 year, but at least you have the information in 1 second. You can drill down into what was found, and it gives you a good overview of that you can then work further on. Okay, so of course you can try it out. So if you go to tripdatabase.com, there's evidence maps up there, and you can try your favorite disease to see what comes out. So I've been talking about how medicine is done at the moment with these clinical trials. The problem with pretty much every clinical trial is that there are very few people involved. So here's another one which has to do with radiology, CT, images, 1,202 people. Well, it's more than the previous one, but it's still very little. So if you just put this into perspective. So let's say that is a clinical trial for radiology, maybe the 1,202 people, and that's what's looked at. In fact, this is really small because in one department in one hospital, they have, it's a really tiny part of the more than 4 terabytes of images that they collect in 1 month, which even more is a really tiny part of the greater than 1 million CT and MR images that are captured in the EU in 1 year. So here the question is, the clinical trials just look at a sample of the population. What if we could make our medical decisions based on all of the patients in all of Austria or all of the EU? What could we do with that? And there's a lot of data out there. So the data is there, not always easy to get, but it's there. So for example, the radiology departments store all of the images that they've taken. These images are also connected to text information, like radiology reports, where radiologists have described what's in the images, and they could also be connected to patient records, they could be connected to lab results. So there's huge amounts of information there. And this is where the company ContextFlow comes into it. I'm a co-founder of ContextFlow. ContextFlow is a spin-off of an EU project that ran from 2010 to 2014, where we were looking at search engines for medical information. And it turned out that this use case for radiologists was very promising. So currently, we're trying to take huge amounts of radiology information into account to assist radiologists. So let's look at that in more detail. There you have a radiologist, what radiologists do is they spend their days sitting in sort of darkened rooms looking at 3D CT and MR images. So they scroll through the various slices of these images. In total, sort of, well, let's say on average, if you just look at these slices, they look at 45,000 images per day. And what they need to do is they need to discover the anomalies in the images. So where in those images are patterns that somehow look strange? And once they found that, well, either they know what it is, but on average, again, in 20% of the cases, they're not sure what it is. There's some uncertainty. And of course, this is higher for younger radiologists than those with more experience. And then they have to find out what it is. So currently, what do? They either ask a colleague, they might look in some books, very often they go to Google. And again, on average, it takes 20 minutes to go from having this uncertainty to actually coming up with a good hypothesis for what this is. So that is a long time to find and analyze the information. So what we're doing at ContextFlow is creating an assistant for radiologists. So the assistant looks like that. So basically what it allows the radiologist to do is, if the radiologist sees something that looks strange in the image, to basically draw a box around it. And then the system goes off, and we have a collection, at least for the lungs, of a few thousand images where we know what the images, what the person has. So then it does a visual similarity comparison to find other images with bits that look like that, brings them back like a search engine. Because we know what these images have, we can then create the histogram that you show at the top that makes some suggestions to the radiologist as to which particular diagnoses occur most often in those images. And given that, we then also go to other information sources, which you see over here. So here we go to Radiopedia, for example, which is a well-known, well, basically Wikipedia for radiologists on the internet. And we also have a cooperation with a medical publisher, TIMA, so we can bring back information from them. Also differential diagnosis. So again, within 1 second, the radiologist has all of the information that he or she needs to be able to make a hypothesis or come to a potential diagnosis and come to the decision. So this saves all of the work of having to first find the information. The information is immediately there. Now, one of the really difficult things about this is that we're working on these large 3D images. So doing the similarity comparison between a few thousand images is really tough. So back about 8 years ago in the EU project, the first prototype of this needed about 1 minute to do a search. So you had to press the search button and wait 1 minute for it to search through all the images and bring it back. So we've got it down now, after a lot of work, to less than 1 second. So the important capability that we have there is this ability to compare images really rapidly. And this opens up all sorts of possibilities. Because we can cluster patients. So we can take groups of images and actually create these clusters of similar patients. And potentially we can then look at treating these patients in a similar way. But of course, this only really becomes interesting when you start taking the time aspect into account. So patients go through hospitals, they have various treatments done, they have radiology images taken, and the patient records written. So if you sort of see a patient trajectory as being a line that goes through some hyperdimensional space, and you take all of the data for the patients and put all of those patients into that space, you're going to be able to find what we call over here the trajectory bundles. So those are the patients that are very similar to each other. And if we can then find these similar patients, we can know which treatments they've had. And if we find a patient that sort of belongs to a bundle, we can make a prediction about that and potentially use that to decide on which treatment to give. So this is where we're going with medicine. So how medicine's becoming data-driven and patient similarity-driven. Now, as I mentioned, it's often very difficult to get hold of this data. So we at ContextFlow have an agreement with hospitals that provide us with anonymized data, but still it's difficult very often to get access to this data because there's usually the argument from the side of the hospitals, privacy, GDPR, we can't give you the data. But it's then interesting that there's some ethicists who argue from the other direction. So there's this book from 2011 where they say, well, basically in some cases it's unethical not to make the data available for these sort of analysis, because if you don't make it available, people are going to suffer. So yeah, this is becoming more and more known, and I think very slowly there's been an openness to having the data analyzed in this way. So there I'm coming to an end. I'll just summarize the two questions that I at least spoke during the presentation. The first one is, what if you could do systematic reviews in 1 second? Well, you can do a sort of approximation of a systematic review in 1 second. But basically what's being done here is to look at the way that medicine is being done at the moment and to try and accelerate that. So systematic reviews are done. How can we do them faster? This is nice in that it's easier to get people to accept it because this is what they're doing anyway, and now you're offering to do it faster. And the second question is, what if you could take every patient into account for diagnosis and treatment planning? So this is where the big revolution in data-driven medicine is coming. So this is the part that's still difficult to convince people that this is possible and that this brings something. So the big revolution in how medicine is being done, and yeah, if you want to take part in that revolution, so at ContextFlow we're hiring. So please speak to me if you're interested. Thank you.
Thank you, Allan, for this great talk. And we have a set of questions, so I just go on and ask them. So the most popular, how do you analyze medical images using convolutional neural networks or something else? Also, which algorithms do you use for clustering patients?
Okay. So, yeah, it's difficult to answer questions like that because, of course, these are the company secrets. But yeah, of course, we use deep learning, yeah, I can't answer questions like that. I'm always worried I'm going to say something I shouldn't.
Okay, but you use deep learning, and this is what makes the company hipster.
Yeah.
Yeah. Okay. Let me just check because questions are popping in. Is there a formal alliance or lobby for opening data for furthering medicine?
Yes, there are many organizations that try and do this. So there's a sort of informal group in Vienna, especially around the Complexity Science Hub, where we are trying to convince people to open medicine. There's been a lot of progress in Austria. So for example, there's some papers that have been published by people at the Complexity Science Hub using anonymized social security data from Austria in order to make predictions about disease evolution, particularly for comorbidities. So if somebody got one disease, what is the probability that they get another disease? So of course, all the diseases that people have are in their social security records, so they can do useful things with those. And also, especially in the UK, I've noticed there are a lot of initiatives to open data in this way. So there they also have the possibility that hospitals open the data, and that under really high security conditions. So you go into a room, you have to give away your cell phone, any cameras, any electronic equipment. There's a computer in the room. You're filmed while you're using the computer. I don't know if they record what you say, but yeah, so you're monitored, and on that computer, you're allowed to access the data and run your algorithms. And I think then they also check what you take out. So, but that's in particular for accessing non-anonymized data. So, of course, non-anonymized data is really nice, because once you anonymize data, you immediately lose information. But in order to do this with non-anonymized data, you need these high sort of privacy requirements.
Okay, so there is a related question. What do you think about data privacy in this field? So what's your personal opinion on these methods?
Well, I agree with the arguments that the ethicists are making to say that you need to look at if you're not making your data available, is this causing somebody else to suffer? And then you need to weigh that up.
Okay, thanks. And let's say, so here is one. Just to say congrats, this looks awesome, Allan. Hugs from Belgrade. Okay, there are a few more, and we still have some time. So, amazing results. How large are the projects you mentioned? People, effort, time.
Okay, so, well, the TRIP database is very nice and easy to work with, because basically there are two people. So there is the boss and the engineer. And the boss tells the engineer what to do. And then, so getting the data there was quite easy. So we basically said, we need the data. And the boss told the engineer, please send them the data. And we got it. Never works like that with big companies. So, yeah, and then on our side, we had one PhD student who was working on the extraction. And we just got funding to continue that. So that's going to be nice. And the company, so Context Flow, we currently have 10 people. But we've been working on this particular topic since 2010. So it started as an EU research project. And so, yeah, already there. Yeah, so basically it's taken a long time to get it this far.
And how do you get access to a few hundred medical trials? A few 100,000, you mentioned. So
they're published.
They're published and
yeah,
accessible
so
for free.
You can go to, well, they're not all available for free because of the paywalls, but many of them are open access, so you can download lots of these papers describing the clinical trials.
All right.
Of course, there's hidden information there because the paper always summarizes the clinical trial, and then there's a document that's not published, which is called, I think, the Clinical Trial Summary Report, which is sort of 100 pages or more, which has all of the details about the clinical trial. And then these 100 pages are reduced to the 8 to 10 pages which are then published. So in doing that, there's also a lot of information that gets lost. So there's also there's a lobby at the moment to try and get these clinical study reports released, or the full 100-page documents. I think, again, this opens up the problem. So if you have these, then there's the big problem of reading them all. And there you also need some sort of support from electronic tools to help you get the key information out of those.
Okay, thanks. So I think the next question is about the image similarity detection. Are you also working towards detecting the anomaly in the first place?
Yes. So this is a new feature that's come up. So instead of asking the radiologist to select the features based on the data that we have, we already suggest some things that the radiologist might want to look at. So here's some things that you might want to look at first before you scroll through the whole image.
Okay. In which area are your results used the most?
Well, for the context flow, of course, it's used in radiology because that's where we're working with radiologists at the moment. And it's nice to work with radiologists. So many of them are very open to new technology. And currently also in radiology, there's a huge push towards artificial intelligence. So currently there's, in many countries, a shortage of radiologists. So there are newspaper reports, like in Scotland, that there are a few hundred cases that haven't been looked at by a radiologist yet for over a month. So this looks bad. So that's why there's push in many countries to automate, let's say, currently the more boring things that radiologists do, to make use of them for what the, things that the computers can't do.
So what did take more time while developing your system? Was it data preprocessing perhaps, or developing machine learning algorithms?
Yeah, so, well, the two systems that are shown. So let's say for context flow, well, as I say, this was something that we've been working on since 2010, so it's difficult to say what took more because, but basically, I think what took the most time was actually getting this retrieval time as down to an acceptable level. And in order to do that, I think there were about 4 prototypes or so built over the whole. So 3 of them were built in the EU project, which was nice. You have research funding to do that. So you always build one, discover what's wrong with it, throw it away, build another one with making sure you don't do the same things wrong again. And yeah, after that there was a 4th one built as the commercial product.
Okay. So last question perhaps. Let me just find it. Well, what is your favorite success metric in the context of project? It's like true positive rate, false negative rate.
Yeah, well, they're basically the retrieval metrics. So we want the correct case to be as often as possible in the top 10, for example. So precision of 10 would be a good metric.
Okay. Okay, any other questions here? Perhaps the last one?
Yeah, I have one. With the systemic review, isn't there a chance that you carry over the problems which are in the research world apparent? Like, for example, this researchers p-value tuning, because the positive prediction value, for example, it never says that most of these hypotheses, they are wrong in the first place. And if you then tune it up, don't you carry over the mistakes which have been made by the researchers because the publish or perish sword which is hanging over their heads?
Of course. So that's why I'm also pushing this move towards the data-driven medicine, because there are all sorts of biases in the way systematic trials are done. And the biggest one is the publication bias, because it's more likely that a trial that has positive results will be published. So I also mentioned that when we did our sentiment analysis of the conclusion. We found most of them, I think if I remember correctly, something less than 10% of the conclusions had negative results, because people just don't publish the negative results. But I'm sure in real life, there are many more negative results than positive results. But the negative results disappear into the bottom drawer and they never get published.
So there should be a push for publishing negative results as well.
Yeah. But that's the problem with the scientific system. Yeah. So people don't get, let's say, recognition for publishing negative results.
Okay, thank you, Allan. So for the rest of us, I'll see you guys in the Zagreb stream.